Assalamu alaikum, everyone. In the previous lecture, we started discussing cloning, and towards the end of the lecture, I summarized all the steps of cloning. And just a quick reminder, cloning requires the use of plasmids, and these plasmids could be cut with a restriction enzyme, and the DNA to be cloned must have sticky ends that could be generated by cutting the DNA fragments to be cloned with the same restriction enzyme. So now we have sticky ends on both ends of the DNA fragments, and these sticky ends are compatible with the sticky ends generated on the plasmid. Another enzyme called DNA ligase can make phosphodiester bonds and can join the inserts with the plasmid like this. And the result is having a recombinant plasmid. But sometimes the ligase, because both ends of the plasmids, when they are generated by the restriction enzyme, both ends are also compatible with each other, which means that they can base pair with each other, and the ligase can convert the plasmid back into its circular form. So we have self-ligated plasmid. The next step is to transform the recombinant plasmid or whatever is produced after the ligation step by the next step is to introduce these plasmid into bacteria so that we have replication of these plasmid inside bacteria. Now, some bacteria might take the recombinant plasmid containing our DNA fragments that we want to clone. Some bacterial cells, they might take the self-ligated plasmid, and some bacteria, actually, a lot of bacteria will not take any of these plasmids. This process is called transformation. There are other techniques that could be used to introduce plasmids into bacteria. Now, when we do cloning, we want the bacteria to have the correct recombinant plasmid. And this bacteria is called transformant bacteria or recombinant bacteria or recombinant cells or transformant cells. Now, next, it's our job to make sure that the bacteria will have our, or it's our job to find the bacteria that will have our insert carried by the plasmid. And this could be easily done by, for example, by taking the plasmid out for analysis. So we could isolate means istikhlas, isolate plasmid and analyze the plasmid that is isolated. So we could do this using a technique called gel electrophoresis. So gel electrophoresis is a technique that could be used to analyze plasmids or to analyze any DNA. So I want you to understand that when you have a plasmid, okay, and this plasmid contains, so this is a circular plasmid. And this circular plasmid contains, for example, a restriction site. Let's say for EcoR1, the enzyme restriction enzyme EcoR1. And this restriction site can be recognized by the enzyme, and the enzyme can cut the plasmid here. So if we take the plasmid and we cut it with eco R1, for example, we will have now a linear plasmid. Okay, so this is a linear plasmid. Okay, and here we have the sticky ends that are generated by cutting this plasmid. These are what we call the sticky ends. Okay, so a single resection site on the plasmid means that if the plasmid is cut with the enzyme, now we have a linear plasmid. So this is a linear plasmid. I draw it like this just to indicate that it's a plasmid, but actually this is a linear DNA molecule. This is a circular DNA molecule. Now, when you add, for example, an insert that has already also stick ends like this, equal stick ends, for example, and you add it to the plasmid, okay? So add to the plasmid, plus, of course, with the help of the DNA ligase enzyme, what will happen is that now we will have the following. We will have a plasmid. Let me make it bigger. Now this plasmid will contain the insert, okay? So this is the insert to be cloned. This is the DNA that we want to insert into the genome, into the plasmid. And also the plasmid will now have two resection sites for EcoR1. So here we have EcoR1 restriction sites, this one and this one. So we have two. So after ligation, now we will have two. Why? Because here we have stick ends. With these stick ends will form this restriction site. Stick ends with this stick ends will form restriction site. Now, this is the recombinant plasmid we have been talking about. Okay. Now imagine if we take this recombinant plasmid and cut it again with EcoR1, what will happen? So now this recombinant plasmid, if we cut it with the resection enzyme EcoR1, what what do you expect will happen? Anyone? So how many DNA molecules will be generated? And what do you think about these DNA molecules? Are they going to be linear or circular? First of all, how many DNA molecules will be produced, will be generated? Two. Why two? Ya Shahid? You are right. Two. What are these two? You will have the plasmid, a linear plasmid, okay? And then you will have the insert, the linear insert, okay? And both, they will have stick ends, like this, okay? Because the enzyme will cut here and here, removing this one, which is this one. So this is the insert. The insert is the DNA fragment that we want to clone, okay? And this is the linear plasmid. Linear means خطي. Okay? Now, imagine if this circular plasmid consists of 5,000 base pairs. So BP indicates base pair. Okay? Imagine if this one is 5,000 and our insert consists of 1,000 base pair, which means this, what is the size of this recombinant plasmid after the ligation of these two together? What is the size of the recombinant plasmid? 6,000. Yes, you are right. So this one recombinant plasmid will contain 6,000 base pairs. What about here? If we cut this recombinant plasmid with EcoR1, what is the size of this DNA molecule? What is the size of this DNA molecule? Of course, these are all approximates. Okay, so approximately, let's say. This one will be 5,000. And this one, probably 1,000. Okay? Good. Now, let me go back to this slide. We did the restriction digestion. So this step is called, this is restriction. When you cut the DNA with an enzyme, this step is called restriction digestion. We do the ligation reaction with the DNA ligase. So here we have ligation, followed by transformation. And now my plasmid inside the bacteria. And because we have millions of bacteria, we need, again, to determine which bacteria contains my plasmid, and the plasmid is carrying my insert or the DNA that I cloned. So I want this bacteria. I don't want this bacteria or this bacteria. So one way is to take the plasmid from this bacteria and take the plasmid from this bacteria and analyze the plasmid by a technique called gel electrophoresis. So before I show you how we could analyze these plasmids, so one way is to isolate plasmids and analyze by gel electrophoresis. So let's discuss what is electrophoresis, and then we'll come back and see how gel electrophoresis will allow us to determine which bacteria will have the correct plasmid. So gel electrophoresis, from its name, if we analyze electrophoresis, so it will involve the use of a gel material, okay? And this gel material is made from a substance called, a sugar called agarose, okay? So agarose, this is a substance, usually it's a powder. We buy it in the powder form, and we add it to a solution or a buffer, and then so we have basically a powder, and then we mix this powder inside, we add this powder inside a buffer. We heat or boil this buffer and the agarose powder, and now we have a solution. And then this solution will be hot, and this solution, we take it and we pour it we pour the solution into what we call a gel cast. A gel cast, this is a gel cast. So this is a chamber or a cast that has a rectangular shape. So this is the shape of the gel. We prepare this gel like we prepare Jello in our homes. So you buy the Jello, el Jello اللي بناكله. You buy it in the powder form and you mix it with water, then you have to boil it, then you have to cool it. Then you have to pour it into a dish, and the Jello eventually will become semi-solid, okay? And it will take the shape of the dish you pour it in after you cool it, okay? So after we pour it into a gel cast, we have to wait and cool it. So the gel will take the shape of this gel cast, okay? And now we have a gel. And in this gel, we have these basically small rooms or chambers called wells or small wells, abar. So this is another picture showing the gel after cooling it. Okay. So this agarose sugar, it's a natural sugar found in seaweeds. Okay. الأعشاب البحرية. Now, this gel allows us to separate DNA fragments and analyze DNA fragments, or allows us to look at the DNA. So the gel here, as you can see, it's a slab of a gel with holes in it. These holes are called wells. Okay. And this is where we add the DNA. So the DNA that we want to analyze, we add it into these wells. Now, we take this gel, we put it inside this electrophoresis chamber. This is electrophoresis chamber. Okay. And when we put the gel, we put buffer inside the chamber. So we submerge the gel and the wells in a buffer solution. So here we have also inside the chamber is filled with a buffer solution. Okay. So this is the chamber. The gel is inside the chamber, and we have buffer covering the gel. This is a side view. So this is the chamber. Okay. This is the chamber. And in the middle, the yellow rectangle indicates the gel. And as you can see, the gel is submerged or maghmoor in the buffer. And here, this is the well. This is a side view of one of the wells. So this is the well that represents maybe this one. Okay. Now, notice that this technique is called gel electro, and we have phoresis. Electro means electricity, kahraba. Phoresis means movement. So now, when we put the DNA here, so inside the wells, we put the DNA will be moving through the gel movement by the action of electricity, by the electrical force. Because as you can see, this electrophoresis chamber has on both sides electrode. One is a positively charged electrode called the anode, and the other charged electrode is called the cathode. And both the anode and the cathode are connected to a power supply that will provide the electricity. We add the samples closer to the cathode, the negatively charged electrode, because the DNA is, what's the charge of DNA? Is it negative or positive? DNA charge, it's negative. Why? Why is it negative? What component in the DNA confers this negative charge to DNA? Yes, because of the phosphatic groups. So now we put the DNA here on the negative side, and now the DNA will be moving in this direction towards the positive electron, okay? Because negative moves towards positive. So Applying electricity will force the DNA to move from the wells towards the other end of the gel or towards the positively charged anode. Okay, that's why this technique is called gel, because the DNA will be moving inside the gel. So as you can see here, when electricity is turned on, the DNA will be moving inside the gel. Okay. So if we put DNA sample here, the DNA will be moving in this direction. Now, the speed of movement of DNA will depend on the size of the DNA. So for example, larger DNA fragments will move slowly, while small DNA fragments will move faster. Okay? So smaller DNA fragments will be moving faster than larger DNA fragments. Okay? This allows us to separate the DNA fragments based on their size. Okay? Now, this gel, you have to understand that the DNA that is added to the wells in order to separate it. So let me tell you something. DNA is colorless. The gel is also colorless. The buffer is also almost colorless. So they don't have color. So we can only see the DNA after. So we allow electricity to push the DNA to move through the gel. Then we turn off electricity and we take the gel out like this. We take it out. And then we expose the gel to UV light. UV light will show us where is the DNA inside the gel. Of course, in order to see the DNA, we need to use a DNA dye. A DNA dye, like we have a DNA dye called ethidium. So ethidium bromide is a chemical that is used to stain substrate DNA, to stain DNA. This ethidium bromide binds to DNA, and when it binds to DNA and the UV light excites this ethidium bromide, the ethidium bromide bound to the DNA will generate this fluorescent lines. These fluorescent lines are called bands. They are bands of DNA. Okay? So each band represents millions of copies of DNA fragments having the same size. So if we zoom in into one of the bands, like this, what you will see millions of DNA fragments of the same size. Of course, we cannot see these each individual DNA fragment. So here we have millions of DNA fragments with the same size. So all fragments in this DNA band, they have the same size. Of course, they are double stranded. I just Don't want you to get confused, but we cannot see them actually naked eye. We can see these DNA fragments as a group like this, as a band. All the DNA fragments here, they have the same size. All the DNA fragments here, they have the same size. Okay, so these DNA bands, these are lines we can see by our own eyes on the gel after exposing the gel to UV light following electrophoresis. But we cannot see the individual fragments with our naked eye. We cannot see the individual fragments or each fragment of DNA within this band. So the DNA fragments here found in this band, they are smaller than the DNA fragments that are found in this band. Why? Because the DNA fragments here, they moved faster than the DNA fragments here because they are smaller. So they moved further from the well. Okay? So after we take the gel out like this, we can look. The gel is colorless. There is DNA here, but we cannot see it unless we expose the gel to UV light. Okay? أشعة فوق بنفسجي. Okay? Now, after explaining the principle of the gel electrophoresis, now I want to go back to this slide. We want to determine which bacteria, because we cannot see bacteria, by the way. I just draw here 3 types of bacteria just to explain what actually happens at the molecular level. But actually, we cannot see these bacteria. All what we can do is take the plasmids from them, and there will be, you have to understand, you have thousands maybe of these bacteria having the same plasmid, and thousands of this bacteria having the same plasmid, and thousands of bacteria that did not take the plasmid like this. So if I want, let's say, if I want to differentiate the bacteria that will have the plasmid from the bacteria that the self-ligated plasmids from the bacteria that do not have the plasmid. Okay? So we use the same colors. We have a plasmid here, and of course, there will be millions of bacteria that have all this plasmid, millions of bacteria. So what we do is that we blindly extract or isolate plasmid from this bacteria. Extract or isolate. Extract or isolate استخلاص plasmid. Extract, isolate plasmid. Okay, so now there are certain ways by which we can extract the plasmid. Of course, we'll not extract just one plasmid. We will have millions of copies, same thing here, and nothing here. Okay? Then what we do restriction. Sorry. We do restriction enzyme digestion with the same enzyme we used for cloning, for example, EcoR1. Same thing here, EcoR1. Same thing here. Well, you have to understand, we don't know when we take the plasmid, we don't know if we have plasmids or not. We don't know if we have the correct plasmid or not, okay? Because these are very small molecules, we cannot see them. And again, here we will have millions of copies. And here we have millions of copies. Okay? Now, if we go back here, we used EcoR1 to clone this insert into the plasmid, which is already also cut with EcoR1. So if, again, if the size of the plasmid is 5,000 base pair, and the insert is 1,000 base pair. So if you cut this plasmid, this recombinant plasmid with EcoR1, what will you get? What type of products will be produced? We already did this here. All right? What will we have? We will have the plasmid, the linear plasmid, and we will have the insert separated from, so we have two fragments. What about here? If this is a self-ligating plasmid, if you cut it with EcoR1, how many fragments will we get? Only one. So the circular plasmid will become linear plasmid. And what about here? Nothing, because we have nothing. If you treat nothing with EcoR1, you will get nothing. So now we take these and do gel electrophoresis. Okay? When we do gel electrophoresis, on the gel, okay, let's call this is bacteria A. This is bacteria, use a more vibrant color. So this is bacteria A, this is bacteria B, this is bacteria C. If we do electrophoresis, and we have 3 wells for bacteria A, for plasmid from bacteria A, or sample from bacteria A, sample from bacteria B, sample from bacteria D. On gel electrophoresis, if we take the plasmids, we digest them with restriction enzyme EcoR1, then we take the digestion products and we run them on the gel. We put them on gel electrophoresis. We do gel electrophoresis. Then we take the gel out and expose it to UV light. How many bands you will see in the lane? So these are also, this is, so from here to here, this is called lane. And from here to here, this is also called lane. خط أو حارة لحارات السباق. Lane. Okay. So in lane A, how many bands do you think A corresponds to the lane of the sample of the plasmids extracted from bacteria A and cut with EcoR1? We will see how many bands. We will see 2 bands. One of them is large, one of them is smaller. What about in B? So what's the size of this band, do you think? What's the size of this band in B? This one is 5,000, which is this one. This one is 1,000 base pair, which is this one. Okay. What about from B? If we extract plasmid from B and cut these plasmid with the EcoR1, and then the digestion products are run on lane B, how many bands do we see? And okay, 1. What's the size of the band? 5,000. So also it will have the same size as this one, and it will migrate or move like this one. What about here? How many bands we will get in lane C? 0, nothing, like this. So now these 2 bands, actually, they are the linear plasmids. So this one and this one. And this one is the insert. This one. So you have to understand, we do not see the bacteria, we cannot see the plasmid, we cannot see the EcoR1, we cannot see these fragments. But because we have millions of copies of plasmids, millions of these fragments, we can see them on the gel as bands. So the gel will tell us which bacteria contains the plasmid. If you don't see any band, this means the bacteria did not take the plasmid. If you see 1 band with a size equal to 5 base pairs, this means this bacteria has self-ligating plasmid. If you see 2 bands, one corresponds in size to 5,000 and the other two has a size of 1,000, this means that this is your plasmid, linear plasmid, and this is your insert. And this is how you make sure that bacteria A contains your plasmid, and now you take this plasmid and do whatever you want to do with it. Okay. So far, we have discussed the cloning. And in cloning, we have a plasmid, and inside this plasmid, we have an insert. We put this plasmid inside bacteria, and what happens is that the bacteria inside the bacteria. The plasmid will replicate itself, making more copy of itself and our insect along with it. Okay, so now we have copies, and you have to understand that the bacteria itself can replicate. Okay, so more and more bacteria will be so the bacteria will replicate itself and replicate the plasmid inside. So we have more and more bacteria that will be carrying multiple copies of the plasmid and, let's say, our enzyme, like this. So this is one way of making millions of copies of your DNA insert. Another way is by using a technique called polymerase chain reaction, okay? Or for short, it's called PCR. Now, this PCR is used to produce many copies of a specific target segment of DNA, okay? So DNA cloning could be used for the same purpose, but also PCR is a much easier and quicker way of making copies of a specific DNA segment or specific DNA sequence. Now, from its name, this technique involves the use of the enzyme polymerase. So this is basically, we could say that this is DNA replication in a test tube. Chain reaction. So the polymerase will carry out chemical reactions by synthesizing new strands. And it is called chain reaction because we have an exponential increase in the number of DNA molecules that is being copied, like a chain reaction. So the idea is that let me clean the slide. So in PCR, one DNA molecule if we start with one DNA molecule, let's say, so this is one DNA molecule. In PCR, one DNA molecule will be replicated. So now we have two. And these two will become four. And the four, so each one, so this one will give two, this one will give two, this one will give also two, this one will be duplicated into two, this DNA molecule will be duplicated into two also, and this one into two. So as you can see, this one into two, like this one into two. Of course, these DNA fragments, they have the same size. I'm just drawing them shorter because I don't have space. This one is a two. So this is what is meant by a chain reaction. A chain reaction means each molecule will be produced from another molecule, but also it will be used as a template to produce more molecules. So this is what is meant by a chain reaction. Okay, it's going like this. We have multiple reactions in chains, and each product is used as a substrate for the next reaction. Okay, so this is what is meant by chain reaction. تفاعلات متسلسلة. All right. So in the PCR, we have cycles, and each cycle, the number of DNA molecules doubles. So for example, in cycle 1, if we start with 1 DNA molecule, after cycle 1, we have 2. After cycle 2, we have 4. After cycle 3, we have 8 DNA molecules. So after each cycle, the number of DNA molecules will double, assuming, of course, 100% efficiency. So we have exponential growing of the number of the identical DNA molecules. All these DNA molecules, they are identical in their length and their sequence. Okay, so this is DNA replication, similar to the DNA replication you learned about in Biology 101. Now, the key to the PCR, the most important component of the PCR, is a DNA polymerase called Taq polymerase. Taq, it's a DNA polymerase, and Taq comes from the word, or 2 words, actually, Thermus aquaticus. Thermus aquaticus, it's a bacteria from which this enzyme is taken, and we use it in the PCR. This Thermus aquaticus, this is a bacteria that can or lives in hot springs, حمامات مياه الدافئة زي الحمة أو معين في الأردن, which means the proteins or the enzymes inside this bacteria they are heat stable. They are not destroyed by high temperatures. Okay. We have other polymerases like PFU. This is from another organism. That PFU, the PFU from Pyrococcus, the name of the organism from which this enzyme is taken, Pyrococcus. So Thermus aquaticus, Taq. Pyrococcus, it's an archaean, it's not a bacteria, which also lives in hot environments. Okay, Curiosis. Okay, PFU. So these are heat stable DNA polymerases, allows the amplification of DNA and the replication of DNA, amplification of DNA at high temperatures. Okay. So PCR reaction components. What do we put inside the PCR reaction? So we have, let's say, we have a test tube. Or let me make it smaller. So we have a test tube. So this is PCR test tube. This is the tube in which we add the following components, PCR components, مكونات تفاعل. Okay. First, we need a DNA template. This DNA template could be, for example, a genome that contains the DNA fragment we are interested in amplifying. Let's say that we are interested in making copies of this region within the genome. Okay, so we want to make, let's say, millions of copies of this DNA or this DNA fragment, or maybe this gene, whatever the case. Okay, so we need DNA template. So the DNA template could be the whole genome. And then we need also nucleotides. Okay, so we add the DNA template, we add the nucleotides. Nucleotides, these will enable the DNA polymerase to synthesize or make a new DNA molecule. So we need DNA polymerase. We also need a buffering system that will provide the DNA polymerase with the optimum conditions. We also need something we call primers. So primers, buffering system, and we have DNA polymerase. All of these components are mixed together in one tube. And these primers are the ones that will tell the DNA polymerase which DNA sequence or which DNA fragments to start amplifying. Okay, so we have here the DNA polymerase will bind to these primers to make more copies of this DNA. Okay, these primers are actually short, single stranded DNA molecules. We will discuss these primers, and typically we need 2 primers. Okay, 2 primers. So we will discuss the role of primers, inshallah, maybe tomorrow. And then we take this, after we mix all these components, we take the tube and we put it inside the PCR machine. Okay, so the tube will be inside the machine like this. So this is the PCR machine. Or it is called actually thermal cycler. Thermal, the word thermal means temperature. Cycler means this device will be programmed to increase and decrease the temperature and repeat this cycle of increase and decrease of temperature. So actually the PCR machine it is actually could be thought of as a heater and a cooler at the same time. So this machine will heat the sample, and also it can cool the sample to allow the synthesis of millions of copies of the same target sequence. Okay? So I told you after each cycle, that's why it's called cycler, because we have cycles. Within these cycles, we have steps of heating and cooling. Each cycle, actually, so each cycle consists of 3 steps. The first one is denaturation. Annealing. And the third one is extension. Okay, so once we mix everything and we put the tube inside the machine, we will have, let's say, maybe between 25 to 35 cycles. Each cycle consists of 3 steps, and each step is done at a different temperature. So denaturation typically done at 59, annealing between, let's say, 50 to 65 Celsius, and extension is done at typically at 72. Okay, so we have 3 major temperatures at 3 different steps, and these steps are repeated between 25 to 35 times. Okay, so we have inside the machine, we have denaturation, annealing, extension, and then go back to denaturation, maybe between 25 to 35 times. Okay. Now, there is an equation that allows us to determine how many DNA molecules could be generated in the PCR. So we have this equation, which is N equals N naught multiplied by 2 to the power number of cycles. So here is the number, the N number of DNA molecules produced after PCR, and N naught, this is the number of template DNA molecules. So in this example, if we start with 1 DNA molecule, okay, so N equals 1 multiplied by 2. So after 25 cycles, we will have 2 to the power 25. And if you take the power of 2 25 times, let me quickly calculate it for you. The number is 33 million 545,432 copies. Notice if you start with one DNA template, after 25 cycles inside the PCR, you will have 33 million and a half, almost 33 million and a half copies produced. Okay? Inshallah, we will continue discussing what happens inside the PCR reaction at the molecular level tomorrow.